He researches how environmental disturbances, including biological invasions, urbanization, and climate change, alter the relationship between soil diversity, biodiversity, and ecosystem health. His research is interdisciplinary, which includes field and lab experiments, mathematical and statistical modeling, and molecular tools. His research has been supported by prestigious institutions such as the National Academy of Sciences, Engineering, and Medicine, and the National Science Foundation. McCary completed his Ph.D. in ecology and evolution at the University of Illinois at Chicago. His postdoctoral training consisted of an NSF Postdoctoral Research Fellowship at the University of Wisconsin at Madison and a Ford Foundation Postdoctoral Fellowship at Yale University School of Environment. So we just had a lovely chat with Matt about all his grants and all the stuff he's going to be focused on for the next 5 years. And Matt, we're so excited you're here. Make sure you unmute as I stop sharing and you share your slides.
Righty. Great. It's such a pleasure to be on the call today. And I was telling Bobby, I've been really fascinated by just how successful this webinar series is. And it's an absolute pleasure to be a part of this and to kind of share my research with you all. And hopefully you would appreciate my perspective on the world, which is really thinking about invasive plants and thinking about it, particularly what their impacts are on the soil ecosystem. The title of my talk is Plant Invasions, Root Traits, and Implications for Ecosystem Functioning. And I will kind of explain my sort of take on their impact through root traits more broadly. Just as a little bit of background, I'm one that really thinks about soil in general. So think about soil ecology, particularly the relationship between soil biodiversity and ecosystem functioning. But one of the things that I've just learned over the course of my career is really these invasive plants that seem to be really having an immense impact. And that's something that is sort of a really major focal point of my research. So with that being said, I think this particular group would appreciate, any sort of American hardwood forest. So this is something that you can probably see up in the upper Midwest, even in the Northeast in this case here. This is a beech maple forest. And, humans, we are naturally drawn to everything that occurs above ground. So there is diversity of plants in the overstory, diversity of plants in the understory. But when you actually take a moment to really appreciate everything that you see above ground, it's actually all the stuff that's happening below ground that really allows for everything you see upright as you see above ground in this case here. So there's some evidence that actually suggests that about 80 to 85% of everything you see above ground at some point was really sort of recycled through the soil ecosystem, with herbivory and other things kind of taking some of that away. But at one point, all of these species you see above ground were sort of uh, related to the things that's happening below ground. Um, that's where my research come in, is to really try to appreciate all the sort of actions that happen below ground to really understand what's really driving these plant community dynamics above ground, and really thinking about it from the point of how invasive plants could really impact that. So as you continue to walk into that forest, if you just kind of take a moment, instead of looking upright, looking at the greenery, the plants above ground, if you take a moment and stop and appreciate the things that's happening below ground, you'll notice there's this very immensely diverse and very abundant suite of different organisms that live in the soil environment. All starting with the leaf litter environment that then leads into the upper divisions of the soil. And if you just kind of just take a moment and just appreciate that, you'll recognize it's actually one of the most diverse ecosystems that we all have in terrestrial landscapes. And there is recent evidence that actually suggests that of all the terrestrial species that we think occur in the world, we believe that 60% of them occur in the soil environment. Thinking about fungi, bacteria, nematodes, mites, and so forth, they create this really immensely diverse system, and they all have a really important role. And so it allows for the system to function when that diversity is really high, in this case here. One of the things I like to sort of communicate on this slide, though, is to just to really capture just some of that very complex and immense diversity that we have in the soil environment. So first and foremost, bacteria, that is going to be one category that is immensely diverse. They have a lot of different functions, but one that we really often think about, especially in the context of plants, they're very important for breaking down complex organic matter into plant available nutrients. As leaf litter falls or even detritus fall, you actually need organisms to break it down in order for these plants to uptake. And they are very important for actually changing those sort of organic forms into inorganic forms that allow for plants to uptake. The bacteria as a category is actually a very diverse group, but also have a very important function. Another category, which would be fungi, we have separate categories from saprophytic fungi. These are sort of free living fungi that actually have the machinery to break down organic matter. Very important, but they also could be a really important food source for a number of other organisms. And then we have the different categories of mycorrhizal fungi, starting with arbuscular, ectomycorrhizal fungi. They form incredibly important mutualisms with most plants. Roughly 80 to 90% of plant species associated with mycorrhizal fungi at some capacity. We know that these fungi offer up increased nutrient absorption from the environment, including moisture, whereas they get carbon from the plant through photosynthesis. And so they really form this really important network in the soil, and they are incredibly diverse in their own right. And once again, they have a really important function. Then we kind of look at other organisms. So we think about centipedes, very cryptic. A lot of people think they're very creepy animals, but they actually are very important predators. They eat other animals that may feed on fungi or bacteria, and so they actually have a really important role from a predator perspective. We have what we call a macrodetritivore. These are millipedes in this case here. These are larger arthropods that actually can chew and break down the organic matter, such as leaf litter in this case here. They are really important for fragmenting that litter that'll then allow for bacteria and fungi to really complete that decomposition process that then allows for nutrients to be released back into the environment. We have daddy long legs, depending on where you are in the US, or harvestmen. These are arachnids, similar to spiders, but not quite, but they kind of form two different roles. One that can scavenge dead material, namely from other arthropods in this case, or they can be predators, depending on the species, another very important group. We then have a number of different spiders that really are critical for keeping these other organisms down in terms of their population size. So we have ground spiders, lignophiid spiders, we have wolf spiders. They all occur on the ground surface and they really like the leaf litter environment. We have gastropods. These could be herbivores, they could be detritivores, very soft-bodied. One of my very personal groups are springtails. They are incredibly cute. They look like cartoons under the microscope, but they actually are very abundant and diverse, and they interact with fungi very interesting and meaningful ways. One of which is that they graze the fungi, and through their grazing sort of activity, they can actually stimulate fungal growth, and so they actually can really just accelerate how nutrients are flowing through ecosystems. Another category are the mites. The mites are incredibly sort of diverse and abundant. They serve as predators. They serve as bacterivores, so they eat bacteria. Fungivores, eating fungi. They also can be pathogens depending on the species, but as a category, they're hyper diverse and super abundant. We have roly-poly, wood lice, another macrodetritivore that can fragment the litter, very important. We have earthworms. They also can be incredibly disruptive. They can really accelerate decomposition and turn over soil and so forth. And so, just as I mentioned, this is just a subset of the different species that occur in the soil. There are nematodes, archaea, there's other categories that I didn't even highlight in this case here. But one that I want to just try to communicate is that this is a hyper diverse ecosystem. And as I also alluded to, they all have a really important function. And so you can envision then that the function of any ecosystem is going to be really critical for thinking about the soil. And that's kind of where I'm really thinking about in my own research program. So with that in mind, we kind of have a very simplistic view of the structure of this ecosystem. it's an oversimplification, but I think it's a good starting point to really build complexity and understanding what are the really important levers for what drives the functioning of the system that's really underpinned by the structure of that. And so, starting with your classic sort of forest ecosystem, traditionally what happens usually around this time of the year, you have litter that is falling to the ground. We kind of think of this as organic matter, but you can more simplistically think about it as litter input. What typically happens is as you get more litter, you get an abundance of microflora. In this case, I think about fungi and bacteria. And as they're sort of more litter, their populations are bigger, you end up with this acceleration of nutrients being released into the environment, which then really have a mediating impact on what type of plants that you have in that ecosystem. You can also think about that microflora really being the foundation for the rest of the food web and thinking about a lot of organisms that feed on the microflora. In this case, I kind of refer to them as primary consumers. This is a silhouette of one of those stringtails I mentioned, these very cartoon-like arthropods that live in the soil. They feed on the microflora, and through their activities, they could really sort of increase nutrients that are being recycled. And then last but not least, we have predators. There's a lot of different spiders, other arachnids like woodlice, harvestmen that actually feed on these primary consumers. And through their own activities, they can really mediate how nutrients are flowing through the ecosystem. And one of the things that we kind of think about with the soil is that we consider this a sort of bottom-up control system, which is that as you sort of shrink the amount of resources through organic matter that goes into the soil ecosystem, you shrink the pool sizes of the microflora, the primary consumers, and the predators, and also slow down nutrients from being released. But then on the flip side, we also think about the soil food web increasing in abundance and functioning as you increase the amount of nutrients that go into that particular ecosystem in this case here. What's really fascinating is that this is a very simple model. We think about, as I said, a bottom-up control system. As you add more nutrients, that increases the pool sizes and the acceleration of nutrients. But if you shrink that, that's going to have the opposite effect. But in reality, it's a very complex system. There are numerous species that happen at each one of these trophic levels. And part of my work is to really try to resolve that complexity. And this is actually very important because we know that in order for these systems to function properly, you need to have a really uh, healthy and functioning ecosystem. But part of the challenge is that we still don't really understand all the different species that occur in the soil, and that lends itself to a lot of, I guess, heartache in trying to understand that. And just to kind of sort of think about that, we have a number of different global change factors that's happened in the world. You think about habitat destruction, that really impacts the type of nutrients that are going into the soil and the resources more broadly. We have invasive species, which I think a lot about, think about invasive plants more broadly. We also have sort of nitrogen deposition of pollution, all of which these can really impact the number and type of resources that go into the soil environment. And if they have just an impact on any one of those trophic levels, one can envision that that's going to really affect the functioning of the entire terrestrial ecosystem in this case here. For the purposes of the talk today, I'll be definitely talking about invasive plants and how they really impact what lives in the soil and what does that mean for the ecosystem structure and function of that particular ecosystem. And so for me, it's really starting at a very simplistic model, thinking about this as a bottom-up control system and thinking about the complexity. But one of the things I like to do is to really think about how resource changes mediated by invasive plants could really influence what lives in the soil and the functioning of that as well. And namely, thinking about the root traits that are associated with these invasive plants. And so there's a lot of sort of real interesting soils recently, just to kind of flag some of the really high impact studies and kind of comments that had come out recently that really is really worried about the soil biodiversity. And as I alluded to, this is a very important system. And there's, thinking about how invasive plants impact them. If we don't really understand what lives in the soil and what really impacts their ecosystems, that's going to have negative consequences for our own management of these systems. And so it's actually really fascinating that there was a really high impact study that came out in PNAS like 2 years ago, and basically all they did was measure the soil biodiversity, and the number that I highlighted before, which is that 60% of terrestrial species occur in the soil. That's where that number came from. So it's just really fascinating to think about our lack of understanding of the soil, that just by being able to quantify what lives in the soil right now gives you a very high impact study in this case. But needless to say, that's something that I'm very interested in to really resolve that complexity and really use invasive plants and their root traits as a mechanism to really understand what structures these ecosystems, and then how does that play into the functioning of these terrestrial systems. So this leads me to then plant invasions more broadly. So plant invasions, typically, we think about them as plants that are overly abundant and can cause some type of environmental or economic damage to an invasive system, in this case here. And largely they are non-native, meaning that they originated from another region. And once they get introduced into a new system, they don't have any predators or herbivores. They may not have the same pathogens. They may have a competitive advantage over native plants. And so what happens, they end up becoming a very strong monoculture where they just basically erode the diversity of the native plants in that given region. There are so many examples to sort of pull from, but Japanese knotweed is one, where basically it just chokes out all the native vegetation. We have invasive Phragmites is another one that happens in wetland ecosystems. We have the comic buckthorn, something that you see in the upper Midwest, even in the Northeast in this case. I actually have a hate for this plant because I studied it as a part of my dissertation. Uh, as the name suggests, it has some very sharp thorns, and it creates these very dense thickets, and it makes 5 feet feel like 50 feet. And it just basically chokes out all of the understory, and it's really problematic once it's invaded in certain forest ecosystems. And something that we definitely have down in southeastern Texas is kudzu, and this is invasive vine that literally just can just overwhelm an entire ecosystem, and you can envision that it's pushing out all the native plants and really eroding the diversity of that system. One thing that is very valuable for us to really quantify or even begin to understand how these invasive plants, for one, how do they invade the system, but then also to try to understand how they impact that system. We kind of think about more simplistically the traits that they have, and so generally speaking, we think about seed size or the amount of seeds that they produce, thinking about how tall they grow or how fast they grow. We often thinking about these above ground traits, and try to use that as a proxy for the level of invasiveness. So when you get into a system, you can measure those. You can kind of figure out exactly how that's going to compete against the native plants in the region in that case. So oftentimes we think about these above ground traits, and they often are reproductive functional traits. I also think about these the leaf traits. So, like I said, how big the leaves are, how fast they grow, and so forth. And it's really fascinating when you actually look at the literature. A lot of our understanding of plant trait dynamics, especially in the context of invasion, thinking about those traits that really explain invasiveness, a lot of that has been really from an above ground perspective. And it actually has informed us a lot. We think about there's a lot of different species we could think about that are very invasive, that are related to producing a lot of seeds, where they grow very quickly. They can basically take up space through the leaf area that they have. And so there's a lot of evidence to show that these above ground traits are actually very helpful for our understanding of what makes invasive plants invasive. But that's just really half the story. And that's something that I hope to convince you all that we should be thinking about their impacts below ground as well. There's a lot of sort of evidence that point to these root traits below ground that are very important for understanding what makes them invasive, but then also thinking about their impacts on that soil ecosystem. And that's something that I will be talking more about for the rest of this talk here. So root traits that I often think about is allelopathy. So this is the capacity for these invasive plants, they kind of secrete these chemicals into the soil that actually can reduce the growth of microbes or other native plants. And so they have a direct competitive advantage. They can change the chemistry. They can actually have higher levels of mutualism. They can produce more biomass. Like all these things can give it a competitive advantage over native plants that would actually allow for it to become very problematic once it is invaded in certain systems. So with that being said, a lot of my work up to this point is really to think about, okay, we know that invasive plants really have a pronounced impact on the ecosystem. A lot of what we know has always been in an above the ground context, but unfortunately, the below ground impacts are poorly described, and that's where my work comes in. And to really think about the invasive root trait effect on soil communities is really important because on one end, we know that these traits allow for them to be invasive, but they also will have really important impacts on the structure and function of that ecosystem. And that's where I like to think about. So this brings me to a very special plant, in my opinion. This is the invasive plant garlic mustard. This is a plant that originated from Europe, made it over to the States in about the 1860s. I started invasive in the Northeast and made its way west into the Midwest as well. And this is a very interesting invasive plant that is very problematic in many different ways. Uh, as you all may know, that it's got this 2-year life cycle, it's a biennial. The first year has this very inconspicuous sort of first year rosettes. Then early in the spring of the following year, usually around April, they boat up to about a meter in height. They go into fluorescence. They create these white flowers. Then later in the season, usually around July, they go into senescence. They set seed and die, and they can produce many seeds per plant. And you can kind of see how small they are. And there's some evidence that suggests that these seeds can remain viable in a seed bank for up to 10 years. What's really fascinating about the garlic mustard, though, is that in the second year is where we kind of think that there's production of glucosinolates. So what happens is that when it goes into senescence, the cell walls break and they release out this glucosinolate, which is supposed to be antifungal. And so there's been a lot of evidence that suggests that when you have garlic mustard, it kills the fungi in the soil, it erodes the diversity. And as I alluded to, that fungi is a major food source in the soil environment. It actually really erodes that soil community more broadly. This is also the same mechanism that allows for garlic mustard to be such a rampant invader. As I mentioned, mycorrhizal fungi is a major mutualism for a lot of plants. So when garlic mustard comes in, it releases this glucosinolates. It then suppresses the growth and diversity of the fungi. And so it has now this competitive advantage over native plants because it has disrupted that mutualism that most native plants in the region have in this case here. And so my particular interest is understanding, well, how does this reduction of fungi mediated by, garlic mustard invasion, what does it mean for the rest of the soil food web in this case? So this brought me out to some work in Chicago near where I got my PhD. And so this is kind of a relatively healthy fragmented forest that you can see in the Chicagoland area. You see there's leaf litter in the background there. There's diversity of different plants, and you don't see a monoculture of really any plant in this case here. So this is kind of sort of what a so-called healthy ecosystem will look like. Although it's pretty fragmented, it's pretty healthy from the perspective of not really being overwhelmed by invasive plants. This is an example of what a garlic mustard invaded site would look like here. And all the white flowers you see, that represents a second year garlic mustard plant. And one can envision that in this area where each one of these plants would be producing glucosinolates that's basically killing the fungi and eroding diversity, one can envision that's going to have a really important impact on the rest of that soil community. And one that I should say that, this is a very important root trait, which is allelopathy, the capacity for these roots to actually open up the cell walls once it dies back. These glucosinolates are released into the soil environment. And basically, it erodes the fungi and becomes this mutualism disruptor. Also, it's going to have this potential impact on the rest of the soil food web in this case. So to really look at this in earnest, we designed several studies. Kind of just present one half of it right now. But basically, we were looking at natural populations of garlic mustard, did some experimental garlic mustard invasions to kind of see how it actually really impacts the soil food web mediated by this reduction in soil fungi. All of to say is that garlic mustard definitely has an impact on the community that lives below ground, and then also changes the functionality of that system. But the way that we were thinking about this is that, typically you have your native plants, you have your organic matter, in this case, litter input. Typically what happens is that the native plants have a positive relationship with soil fungi. As you increase soil fungi, you get more fungivores. So thinking about these springtails, we also get orbated mites, which are fungivores as well. You increase as you get more fungi. And then as you get more fungivores, you get more predators. In this case, this could be predaceous mites. These can be spiders. It could be other forms of arachnids like a harvestman and so forth. So you can kind of see that in a healthy ecosystem that's dominated, this is a forest ecosystem, I should say, that's dominated by microbial, by fungi in the soil, that's going to have a really healthy soil ecosystem. In comes garlic mustard. We know there's a lot of evidence that suggests that garlic mustard really erodes the native plant abundance and diversity, has a negative impact on soil fungi. So by this reduction of fungi, though, you would expect to see a negative indirect impact on fungivores and their predators. So as you have garlic mustard invasion, it basically kills the fungi. It has a competitive advantage of native plants, but that reduction of fungi now means there's no food for fungivores and there's no food for the predators that feed on these fungivores. And this was a model that we were kind of working with in this case here. So to test that, we did this study across 5 sites that span I guess, the northeastern part of Illinois. We had 5 sites that had naturally occurring garlic mustard populations. And within each one of these sites, we kind of did a pair design where we had garlic mustard patches that were invaded in uh, variable levels, some that very, very invaded, some that moderately invaded. But we kind of identified these garlic mustard patches and we then paired them with these uninvaded patches, which we kind of refer to as the control plot. So we had 5 sites, 10 plots per site, 50 plots total across all this. And this was done over 2 years. And effectively, these were like 1 by 1 meter square, so relatively small plots. But we basically looked at the entire 1 by 1 meter plot, thinking about the entire ecosystem. We looked at the plant diversity. We looked at uh, the soil. We also measured the fungi in multiple ways. One in which we did a washing technique. We can measure how much hyphae was in the soil as a proxy for food availability for fungivores. We also did metabarcoding on the fungal communities to actually look at how the fungal communities look in terms of the DNA sequences that they have. And then we did some extractions of the invertebrate communities using Berlese Tellgram funnels. So this is kind of a method that you can use to, it basically heats up the soil. And because of these organisms are very sensitive to drying out, you basically force them to burrow out of the soil. And there's a collection valve, and that becomes the way that you measure their abundance and diversity. And so we did this across all of our 50 plots, and we did this for 2 years and was really trying to understand how does garlic mustard really change the soil ecosystem from that perspective. What was really fascinating is that what we found is that garlic mustard, not only did it change the abundance, we have evidence to show that the abundance of hyphae was much lower in these invaded plots, but we also found that they really restructured that fungal community in this case. And so just to kind of talk about, this is the ordination plot here. And each one of these symbols represent a given community. In this case, the black circles represent invaded plant communities, and the open triangles represent the uninvaded plant communities. And what we found is that the meta barcoding of these fungal communities, so think about the DNA sequences of the fungal communities, were quite different in these invaded patches compared to the uninvaded patches. And what I have here is these vector overlays. So what this shows is that the longer this line is, shows the stronger that relationship is. And this is one of our very most abundant fungivores in our plots, which is the orbatic mites. They most often associated with these control plots, meaning that as you get more control plots, you get more orbatic mites, and they negatively correlated with these invaded patches in this case here. So this is kind of one of the sites that we looked at. This happened across all 5 sites. There were always very different communities of fungi in these invaded and uninvaded patches, and they often correlated with the fungivores. With predators, were a little bit harder to kind of understand in this context here. But I have another measure that actually allowed us to kind of know how the whole soil food web responded to garlic mustard invasion. So this brings me back to we did a structural equation model to really understand how all trophic levels responded to garlic mustard invasion, from the plants to garlic mustard to the fungi to the predators and so forth. And so I'll kind of walk you through this very simply, but this is once again the model that we're working with. So we have native plants, we have our organic matter, and what we found is that there is a positive relationship. So this is a correlation coefficient. The closer that number is to 1 shows that it's a very strong relationship. The closer that number is to 0 shows a very weak relationship. And also have these arrows to denote the significance level. Um, this shows it's a positive relationship between native plants and the fungi in this case, measured by the amount of hyphae we found in the soil. It's not a very statistically significant or strong result, but it does show in the right direction. However, in our plots, what we found is that as you increase the amount of fungi, you get more fungivores. And this was a very strong result here, showing that there's a positive relationship between the amount of fungi and the amount of fungivores you get. And then you see that as you get more fungivores, you get a very strong positive relationship with predators in this case here. So this is actually showing exactly what I mentioned, which is a bottom-up control system that as you increase these trophic levels, they all increase simultaneously in the right direction. In comes garlic mustard. We found that there's a negative, strong impact on native plant abundance in this case here. We also found a very strong negative impact on the fungi in this case here. And then what we found is that basically, because of garlic mustard reduction of fungi, you get this indirect negative impact on the fungivores and their predators in this case here. I will say when you're looking at indirect effects in structural equation model, you can't really measure that statistically. But what we can see is that these coefficients suggest that they are very strong effects when you think about the indirect effects in this case. And so we basically concluded that, by garlic mustard reducing the fungi in the soil, that is really having this cascading effect on the rest of the soil food web, in addition to displacing these native plants. So the summary of this first part is that plant invasion can definitely have a pronounced impact on these soil communities, definitely can change the soil food web structure, in this case related to fungal resources. And as I mentioned, that these effects have this sort of indirect effect on plant abundance. So as I mentioned, as garlic mustard gets into the system, reduces the fungi because fungi is a major mutualism for native plants. It actually has this competitive advantage, and then it leads to displacing the native plant, both in abundance and cover. So this work then kind of stimulated other ideas of like, well, this is just one root trait in garlic mustard as an invader plant. Garlic mustard has this unique allelopathic trait that secretes glucosinolase that suppress fungi. This then begs the question like, well, are these invasive plants that we see in our systems, do they have very different root traits? And then if so, what does that mean for the rest of the soil community? And so this kind of led to a collaboration that I have with a colleague, actually at my former alma mater, University of Illinois, Chicago, Dr. Gabi Nunez-Muir. She's a macroecologist, so she really think about large scale processes. Unlike myself, I think about these very local scale impacts. And so it was a really interesting collaboration because we could kind of blend our 2 ways of thinking. She's looking at broad scale patterns. And I think about mechanisms at the local scale. And so we were able to kind of bridge these two worlds in a way to really understand, for one, does the invasive plants have different root traits? And then what does that mean for the rest of the soil community in this case? And we were looking at this across the entire continental, well, not just the continental, we actually looked at the entire uh, US, including Alaska and Hawaii and so forth. So this was a very large data set. So we basically compiled data from the NEON, which is the National Sport. Um, I forget the NEON. Yeah, the National Ecological Observatory Network. NEON. It's funded by the NSF, and they basically have uh, 50 plus sites all across the U.S. and they have a very uh, cool data set because they basically quantified these plots and they looked at it for multiple years and they have data sets that's looking at native plant abundance, invasive plant abundance. And then what we also did is that we paired that data with root trait data from multiple databases, such as the Global Root Trait Database, and then they also had the DI data set had the microbial composition, which is measured by phospholipid fatty acid analysis, which is a technique that basically looks at what type of lipids you have, and you can kind of know if it's a bacteria, if it's a fungivore, is it a fungi, and so it gives you all of these different lipids that you can then quantify the composition of that microbial community. So needless to say, we ended up with many sites, and we ended up with a data set that included 94 invasive plant species, 632 sampling plots, and this was done over 5 years. And so this was a massive data set that had all these different components, and they were matched temporally, but then also spatially as well. And so we can really do some digging and understand how these root traits differ, and then what does that sort of look at in terms of the rest of the soil community. So what was really fascinating is that the invasive plants definitely had different roots, but it didn't occur across all traits. So in this case, what we found is that specific root length, which is a trait that is measured by basically the amount of like fine root uh, hairs in this, well, not hairs, fine root traits in this case here. So if you think about a plant that has more fine roots, the specific root length would be higher. And fine roots also correlate with being more nutritious, meaning that it breaks down much more quickly, it's not as robust. What we found is that invasive plants tend to have higher specific root length. So they have more fine root traits compared to native plants. We also found that native plants had higher root tissue density, which is actually in some ways not surprising. So this is showing that because invasive plants have higher sort of fine roots, the tissue density for the native plants were thicker, meaning that they have more robust root traits that breaks down much more slowly than these invasive plants. And basically what this is showing is that these invasive plants actually have cheaper roots that could definitely regenerate much more quickly than these more robust root traits of these native plants. We didn't find differences between all these other trait categories, thinking about the chemistry or the root length and so forth. But effectively, what we found is that the fine roots are higher for these invasive plants, and the tissue density is higher for these native plants in this case. And what's really fascinating is that because, and I should mention that these sites occur all over, these are different forests, grasslands, wetlands, and so just by the very nature that on average these invasive plants have higher final roots, what we found is that despite where you are in the United States, if you have a high density of these invasive plants in these terrestrial systems, you get a very homogenized microbial community. It's a little bit hard to see here. The red represents these plots that are heavily invaded with invasive plants, and looking at their sort of impacts on the soil community, microbes in this case. And what this is really trying to show is that the clustering, so as they're closer together, they represent very similar communities of microbes. And as you can see that the red is always so clustered, you can barely even see it in this plot here. As you get moderately invaded, you get more spread out. But then these uninvaded plots have very different communities of microbes. And so what we gleaned from this is that because you get root traits from these invasive plants that are basically have higher fine root traits. They basically end up with a soil community of microbes that are very similar, and it sort of really structures communities that look very similar, regardless of where you are in the United States. And because you get the high variation of these native plants, so these uninvaded plots have a huge diversity of plants. You just get a wide array of different microbes that occur in those soils. And so when we looked at that, we found that basically these highly invaded soils look quite different than these uninvaded soils. And you get a very homogenized microbial community when you have these high levels of invasion. And it's related to the root traits that's associated with these invasive plants. So to summarize, we basically found that native plants and invasive plants have very different root traits, and it's really related to key traits of like specific root length or fine roots, versus the native plants have higher tissue density, and I just mentioned that there. And what we found is that these microbial communities in highly invaded plots were more similar in composition across all ecosystem types and geographic regions than any of the plots with higher proportions of native plants. And so to us, this was really striking. So you can be in Washington State or Florida, and you can see that if you have high levels of invasion, those communities are going to look very similar from a microbial perspective. And that was really fascinating to see that. So one thing I should say as well that these invasive plant root traits explain twice as much variation in the microbial composition community. So basically, what we found is that in these same plots that the microbes occur, you can understand more about the microbial community by understanding the invasive root traits than what happens with the native plants in this case. All right. So very quickly, I'm going to spend just a little bit of time. I realized in creating this talk that I was thinking in the more negative sense, and I wanted to try to kind of leave with an uplifting story, which actually takes me back to some of the first research that I published. But it's really related to, well, we know that these invasive plants have really important impacts on these ecosystems. One of the things I was always interested in is like, well, if you manage these invasive plants, can you restore the soil community, and namely think about the soil and vertebrate community in this case here. So this is going to be some work that I presented that I've done many years ago now, but it's still one that resonates true to this day. So basically, what I did in this experiment, with this natural experiment, where we had sites that were scattered all through the Chicagoland area. We had three different categories. So we had our degraded sites, that sites that have never been managed before. They were overrun with invasive plants. I mean, they had a lot of buckthorn, a lot of garlic mustard, multiflora rose. I used to hate working in those sites. And basically, we wanted to kind of see if these degraded sites look different in terms of the soil arthropod community across sites that have been intermediately managed. So these are sites that have been vigorously managed, but not quite at the reference level. And then we have five sites that represent exactly the targets that we're looking for. Very healthy, high diversity in the overstory and understory, no invasive species, a very healthy leaf litter layer in this case here. And this is really sort of important to think about because oftentimes we think about managing invasive plants is often like with the idea of managing native plants. We think about if you manage the invasive plants, the whole ecosystem will restore. But in reality, there is really no evidence that suggests that these arthropods, which are really critical to the functioning of the systems, if they actually will recover as well if you remove these invasive plants. And so the study was kind of designed to really look at that in more detail. So needless to say that I collected like almost 55,000 arthropods across this study, both in across these degraded plots, these intermediate plots, and these reference plots, and was trying to see if the vegetation itself and the restoration of these communities, thinking about the invasive plant management, does it restore these arthropod communities? So we had a pretty healthy sample size to sort of measure that. And what's actually really fascinating is that, in fact, what we found is that in these restored ecosystems, so thinking about the reference category, we found more native intimate bride sort of springtails. These are incredibly abundant. They kind of walk and they serve as fungivores, as I mentioned, but they also are an important food source for the whole food web. What we found is that they often associated with these reference sites, and they were far more abundant. It had very healthy leaf litter in this case here. And what's really fascinating is that in these control plots or degraded plots, we found an overabundance of this invasive isopod that was all over. And so, what we found in this research, though, is that if you just restore the plant community, we also found that you restore the soil arthropod community with more native entomobrides compared to having an abundance of invasive wood lice in these degraded plots in this case. And this is just more evidence to show that. So, oops, sorry, that the entomobride, what we found is that you just get so many more in these restorer plots, and they were hyper abundant, whereas they were almost non-existent in these control plots. Whereas you can see that this white color here, they were always more abundant in the control and intermediate sites, and they were like almost non-existent in these reference sites. In this case here, really pointing to that these restored communities have a higher abundance of these sort of native intima springtail species compared to these invaded plots. And what's even more fascinating is that we were looking at the composition of the plants or the arthropods, I should say. And what we found is that it's the invasive woody plants that really drove that relationship. So effectively, what this is actually suggesting is that as you increase the number of invasive plants, woody invasive plants in the ecosystem, you get more invasive isopods. Whereas over here, you get the negative correlation in these reference plots. So as you move from the invasive to the more healthy ecosystems, you end up with fewer invasive isopods, but you end up with also less invasive woody plants as well. So really pointing to this idea that it's the management of these invasive woody plants that in theory should be able to restore the soil arthropod community in this case. So to kind of put it all together very quickly, management certainly has a positive impact on the soil community. It's really managing this invasive woody plants that seem to be the driver for these soil arthropod communities. And so, sort of the recommendation we make is that restoration management of these arthropod communities, we need to think about managing these invasive woody plants, and particularly in metropolitan fragmented woodlands. That would be very helpful for restoring the soil arthropod community. I talked about a lot, but I just want to leave on just a recap very quickly. So, kind of led with a story of just how garlic mustard and this really interesting root trait of allelopathy, how does that sort of impact these soil food webs? And so we found that by this reduction of fungi, it actually does restructure the whole food web that then leads to this overabundance of garlic mustard in these plots. And that can erode the diversity of those particular ecosystems. And with that in mind, is that's just one example of garlic mustard. But when you actually scale up and look at across all invasive plants, we do find that these root traits are different for these invasive plants, and that the differences in these root traits actually have a really important impact on the structure of the microbial composition. In addition to probably thinking about the entire soil food web, we didn't get a chance to do that in a study there, but we can now say that invasive plants do have different root traits and that they also have a really important impact on the soil ecosystem. And then to kind of leave on a more cheery note is that despite all of that, what's really nice though is that if we can manage for these invasive plants, and hopefully through time, we can actually restore these soil arthropod communities in addition to the functions that they provide for these ecosystems. So with that being said, I just will acknowledge all the great people kind of presented work that span parts of my career. I want to thank all of my advisors, postdoc and Ph.D., my current lab, all the folks I worked at the University of Wisconsin. And with that, I would be very happy to take questions.
Hi, Matt. I felt like I was back at school with one of the great professors.
Thank you.
A
I
little
appreciate that.
overwhelmed, but catching it. We have a ton of questions. I'm going to try to group them a little bit together. Um, One of the questions was, are there invasive bacteria, invasive microbes that come with these plants? Like,
Yep.
so, yeah, okay. So tell us
No,
about that.
yeah, that's actually a really interesting new avenue of how we understand invasion as well. It's like co-invasions. And so some of these invasive plants cannot persist on their own. The ones that come with their co-invaders and like microbes or think about bacteria, even fungi for that matter, uh, that allows for them to be invasive. And actually, one of my colleagues at the University of Houston, right down the street, has published some work that shows that without their mutualists, they aren't invasive. But when they have the co-invasion of their soil
Oh,
mycorrhizal fungi and the invasive pine in this case here, it basically takes off and become really problematic. So yes, they certainly are invasive microbes that are also really problematic and can help facilitate the invasion for some of these plants.
that's really interesting. Couple questions about the garlic mustard. Understood that yes, you focused your research on that, but can it be used as a broader model of an invasive plant that garlic mustard might come in and then go away, although it's changed the soil? Does it then
Yeah,
open up for other things? Somebody mentioned mugwort came in after
yep,
their garlic mustard. Was that
yep,
because of the change in the soil? Can you explain some of
yeah,
that transitioning?
definitely. Now, garlic mustard is an interesting one because of the allelopathic sort of trait that it has, and so uh, unfortunately, it's very hard to get back to pre-invaded soils when you have garlic mustard invasion, and so there's even some evidence that suggests it takes about 6, 7, 8 years before the microbial community to restore or get back to what it was pre-invasion. And it just does so much damage to the soil environment that I think it actually would facilitate other invaders. And what's actually interesting about garlic mustard is that long-standing garlic mustard patches start to weed out. It starts to lose its rigor, but then it may actually facilitate the invasion of other plant invaders that could then outcompete it. And so I think that I'm not surprised to hear that mugwort or some other invader. I know Japanese steelgrass is another one that people mention that can come in
That's
and
my house.
displace. Yeah, so it's but I think it does so much damage to the soil that it then makes it possible for other invaders to come in and become more problematic.
So you're saying the soil without any intervention or restoration of the plant material would take 6 or 8 years to revive to become more like it was originally. Is it any faster if you go in and try to use some restoration methods, remove the invasives, and put some healthy
Yeah,
native woody?
that's a great question that I wish I would have done a follow-up. So this is Rick Lankhouse work that I'm referencing. He had done some really cool work that was looking at how long does this impact of garlic mustard last on the fungal community. So when you remove garlic mustard, it can take like 6, 7, 8 years before it restores back to somewhat like pre-invasion levels. I don't know if any research that has followed up on that to see if you can accelerate that by inoculating the soil with fungi or some type of plant material. I don't actually know. I do know that once you have these very long-standing garlic mustard invaded patches, though, it could be really, really hard to get it restored back without. If you just let it go, it's just going to take a long time. So, but it'd be
Yeah.
interesting to see if there is a way to accelerate
Hmm.
the recovery. And I don't know if there's any research that's been done on that per se.
Okay. Um, Another question about the root traits.
Hmm.
So these root traits don't change or evolve when they come here. They were actually a natural feature wherever these plants are from. Is that correct?
Yeah, so that's a really interesting question that I also would hope to be able to investigate in I guess that part of this grant I have, but also think about later on down the road. There are some ideas that in their the native sort of ecosystem, that they still have the same roots, but because they have uh, a very constrained or competition with other native plants, the root traits probably function slightly differently than when they are in the introduced region. That's just speculation. I don't know of any real studies that looked at that outside of the garlic mustard system. So we do know that in general, like garlic mustard, when it's in Europe, uh, the potency of the allelopathy is actually far less, but it's the
Mm,
ones that's
mm,
invaded. It's called the novel weapons hypothesis, that it has this really potent allelopathy in an invaded stage. So we think that there is some change that happens that allows for it to become invasive in this new region. But it's hard to kind of generalize across the board on that one.
mm. So two questions, two different angles, but same question. Is the soil biome impacted by controlled burns of invasive plants? And the other question was in using Roundup um, in
Definitely.
to remove them. So the biome would be changed again by the
Yep. Yep.
Roundup and the burn.
Yeah, so that's another. I got some really excellent questions, by the way. I would say
I'll
that
send them all to you so you can think about them for new research.
yeah, I would say that fire is probably less of a concern, because that is, generally speaking, a more natural process that a lot of these ecosystems, I'm thinking about the Midwest and probably in the Northeast as well, it's probably a more natural disturbance that occurs. So the recovery is probably much more quick. I do
Okay.
worry about like Roundup or any herbicide that probably could have some really important negative impacts that could limit the functionality of the soil. And I do know some colleagues who've done that to show that certain groups aren't as effective at breaking down nutrients in the soil, and that can really impair the functionality of the soil environment. So I think if it's spot treated, that's fine. But if you think about like really large scales of herbicide being applied, I definitely think about the impacts on the soil for sure.
Hmm. Somebody wants to know how they can follow your work.
That is a great question too. Look at that. I'm a little bit of I do like to interact with folks who are really interested in this type of work. I need to do a better job of like thinking about social media and making myself available. I am kind of just naturally introverted. Like I get really geeky when I get to talk about my research, but it is like social media scares me. I should be better about that. I do try to make it accessible. I know there's paywalls on publications and things of that nature, but um, I guess I need to think about that. Yeah.
We have you have your McCary Labs
Yeah, that's
web
true. I
page.
do. I do
That's a
have my
start.
webpage. That's the start. Yeah,
Yeah.
yeah. Check out my webpage. In fact, we are due to update that. I have a lab manager who's responsible for that. So,
Okay.
but I think at some point, my lab is very eager. They much younger and they think about social media. They want us to be more active in that way. But I appreciate the
Mm.
question. I think I should do better
Okay, and I have one more, and we'll close with this. How the heck did you ever decide to study something you can't see?
at that. That is once again a really great question. Honestly, for me, the soil is actually one of those things that has always been really fascinating. And I guess as a short story, so I'm more of a entomologist than I am a plant ecologist, although I really think about plants as a very critical part of my program. But it's really thinking about what these insects do that really got me interested in
Mm.
things that live in the soil, from like spiders. I have tarantulas. They're very cryptic. They have really interesting life histories, and so it's really thinking about these cryptic species that got me interested. And part of it came out of my father, who was a pest control technician, and he used to take me to work with him. And I used to be fascinated by seeing like these roaches that's crawling away from the refrigerator, and just seeing all these cool insects, although pests. And so that was kind of what really stimulated my interest in just understanding their importance in ecosystems. But yeah, I appreciate that question.
Well, thank you so much, Matt, for being with us. You have all sorts of kudos in the in the Q&A here.
That's awesome.
So thank you for sharing your knowledge, your research, and helping us look kind of below the surface.
That's all. This was a lot of fun, and thank you again for the introduction. I think what y'all doing is great, and hope to interact with you all more.
Fantastic. We're here.
That's awesome.
All right. Take care, Matt. Thank you, everybody behind the scenes and
Take care.
everybody who's left on here. We really appreciate you coming tonight. Night.
Thank you.